Hello everyone, welcome back to another session of RNA biology. So we were learning the roles played by non-coding RNAs in various gene expression events, developmental events, and also diseases such as cancer, how they are contributing to various pathways, how they are contributing to tweaking of various proteins and their functions, and eventually it helps in manifesting a given cellular physiology. Sometimes it is beneficial, sometimes it is harmful, such as cancerous condition. So we were here in the previous lecture that the non-coding RNA TERA, the LNC RNA transcribed from the telomeric ends, so which can bind to the telomerase enzyme, inhibiting its activity, and it is downregulated. This non-coding RNA is downregulated in the cancerous cells, because of which the telomerase activity is high, enabling the cancer cells to evade apoptosis or cellular senescence and subsequent programmed cell death. So cancer cells need to have that because they divide rapidly, and there is a good chance that they will exhaust or reduce the length of telomeres much rapidly than a somatic cell, so that make the cancer cell vulnerable to apoptosis. But because of this reactivated telomerase activity, that means the negative regulator of telomerase, that is the TERA, is downregulated in cancer cells, and which will activate the telomerase enzyme, which makes the cancer cells more fit, more adapted, and help them in evading the apoptosis. So what we should understand, that the non-coding RNA helpful in certain biological mechanism will be manipulated in a cancerous condition, and that will make the cancer cells eternal. They can live forever. So now let us see another example, that is P10P1. So that is P10P1 stands for P10 pseudogene 1. So the aberrant activation of PI3 AKT pathway in melanoma, melanoma is a type of skin cancer, is known to be caused by a genomic deletion, promoter methylation, and loss of function mutations of tumor suppressor genes. We know tumor suppressors, we kind of discussed about P53, what are the roles played by P53 in one of the earlier classes, and there are several tumor suppressors are there in our body, and one of the major tumor suppressor is P10, phosphatase and tensine homolog. So P10's role is to maintain the balance between PIP3 and PIP2, phosphatidylinositol phosphate, diphosphate and triphosphate. So it maintains the balance between these two. So this basically a phosphatase, and this balance is important to maintain a healthy level of AKT. So P10, when it is absent or the P10's activity is absent, the AKT level will go up, and that can facilitate the cell proliferation. So AKT-mTOR pathway is an important pathway. So P10, as I told you, is a dual specificity phosphatase. It has got phosphatase and tensine homolog as a full name. So the P10 is a tumor suppressor also, similar to P53, but the functioning is different, not like P53. So the levels of P10 tumor suppressor protein is also regulated the post-transcriptionally, at the post-transcriptional level, by a complex microRNA network involving the long non-coding RNA P10P1. Its name is P10 pseudogene 1. P10P1 behaves as a pseudogene of P10, or we can also call it as a P10 decoy. So pseudogene basically works in multiple ways. When you have a pseudogene for a gene, that means when you call it pseudogene itself says, like if you say iPhone mimic. So when you say iPhone mimic, itself indicates there is something called iPhone Xist. You cannot say this is an XYZ phone mimic, because there is no phone is there with a brand XYZ, right? So when you say iPhone mimic, we should understand, oh, there is something called iPhone Xist, which is having a defined function. So when you say P10 pseudogene, that means P10's function or PTEN's value, somehow the pseudogene can influence. Pseudogene can come from various reasons, like sometimes pseudogenes will have some mutation, it mimics the actual gene but it's not the actual gene. Sometimes pseudogene will have a lot in common to that of the actual gene, but it will not interact with the same counterparts or same proteins what it is supposed to interact as done by the original gene. So this we usually call the pseudogene as a decoy. Decoy means it can fool the system. That means it will look like the original gene, but it's not the original gene. So many times it will act like a scavenging molecule. That means the molecules that interact with the PTEN will get competed out, something like that. So this is the PTEN P1 does a competitive binding of the miRNA that is capable of down regulating the PTEN expression. So what will happen, that the cell will maintain the levels of tumor suppressor protein sufficient to restrict cell proliferation. So what we can understand is that PTEN P1 behaves somewhat like PTEN, but it is not doing exact functions what a PTEN is capable of doing. So even in the absence of PTEN, cells are supposed to be proliferating because it is a negative regulator of the AKT pathway. So if you are not having the PTEN functioning properly, then what will happen? The cell will proliferate. So if PTEN P1 is present, what it will do? It is protein. It will PTEN P1 make sure that it will restrict the proliferation within the limits, but it cannot restrict as much as PTEN is capable of doing. So in many human cancers such as melanoma, they exhibit a loss of PTEN P1. That means in cancerous condition, they have to divide actively, quite solidly, quite strongly. So what actually happens is that the PTEN P1, when it is absent in melanoma, that will lead to a very aggressive cell proliferation and very aggressive cancer. So they exhibit a loss of PTEN P1 long non-coding RNA, resulting in a decrease in PTEN expression and a loss of levels of PTEN tumor suppressor protein is sufficient to allow unrestricted cell proliferation. So what we should understand is that the PTEN decoy, that is the PTEN decoy is the PTEN P1 is absent from the melanoma cells, that can lead to the much prolonged decrease in the PTEN levels itself, because PTEN P1 is acting as a decoy and the PTEN P1 expression is missing, and this eventually leads to decreased levels of PTEN, because what we should understand is their expression, the PTEN P1 and PTEN expressions are kind of monitored and they are linked. So levels of one can influence the level of the other. So in melanoma, what we say, loss of PTEN P1 long non-coding RNA results in a decrease in actual PTEN expression and a loss of levels of PTEN, because which is a tumor suppressor. So the absence of PTEN will cause an elevated level of AKT, and this in turn will facilitate the proliferation. So now let us see how does it work. The long non-coding RNA is downregulated, and normally it should be in a steady state level, and the third situation is a overexpressed situation. The long non-coding RNA is overexpressed. So when you see the RNA levels, when they are changing based on the cancerous condition, the mRNA target gene also will get affected for the PTEN, which is the tumor suppressor. So the decoy and the actual gene will have a strong interrelationship, which will be affected very badly in the case of melanoma cells, but it is influencing the PTEN levels through the P10 P1. So another example we can see about the long non-coding RNA leading to the cancer is MALAT1. What is the full name of this non-coding RNA? Metastasis associated lung adenocarcinoma transcript 1. And this gene have got another name, that is NEAT2, that is non-coding nuclear enriched abundant transcript 2. So the overexpression linked to an increase in cell proliferation. When MALAT1 is overexpressed, it can cause a increase in cell proliferation in colorectal and lung cancer. So MALAT1 localizes to nuclear speckles and acts post-transcriptionally by regulating levels of phosphorylated serine, arginine, and it's in short form it is written as SR proteins, splicing factors. So SR splicing factors are affected in the overexpressed condition of MALAT, and in cancerous condition it is MALAT1 is overexpressed. So MALAT1 interacts with another protein called CBX4, and which is a E3 SUMO, a small ubiquitin like modifier, that is called SUMO protein ligase. So CBX4 is a SUMO like protein ligase, and it's also known as chromobox protein homolog 4, which is a component of PRC1 like complex, not actual PRC1, PRC1 like complex that is required to maintain the transcriptionally repressive state in many genes by mediating the monoubiquitination of histone H2A lysine 119, H2AK 119. So MALAT1 regulates the subnuclear shuttling of CBX between the polycomb bodies and interchromatin granules. So MALAT acts as a mediator of the CBX. So this can lead to a aberrant cell proliferation control, especially seen in cancer cells. So another non-coding RNA that comes into picture is MASC RNA, that is MALAT1 associated small cytoplasmic RNA. So it is proposed that the MALAT1 also encode a transfer RNA like structure generated by RNase-P action, which is a ribozyme. Although how this SNC RNA functions is not fully understood yet, it is proposed probably that MASC RNA may act as a sponge for proteins, preventing them from reaching their structural destination. The structural means the interaction natural destination points, and where it is supposed to be actually present within a cell, or it can also act maybe simply via alerting the cells that the MALAT1 is available. That means it can act as a mimic in the nucleus for their nuclear duties. So many times the non-coding RNAs will act as a decoy for an mRNA. Sometimes this related non-coding RNAs can influence or act like a sponge for their related RNA structure. So this will cause an imbalance. That means it can fool the system. Absence, just like if you see something, there is English, there is a saying also, right, that all the glitters are not gold. So what indicates the glitters immediately give you a connection to that of gold. That is why if you see many times the glitters will be, you won't see usually glitters in some odd color. Usually they will be golden or silver color. Usually you will not, people don't use glitters in all possible colors, because the golden color glitters will have a fascination in humans' mind. It connects into gold. So we should understand non-coding RNA also will fool the system, saying that an absent RNA will be reported as present, but actually it is not present. So the system will shut off the actual RNA which is in need, but it is been fooled. Like people say in some like in some places, like some city walls, I don't know which city, like if the city is not having a proper beautiful structures, etc., so they will paint the walls with nice beautiful building structures. So the existing wall will appear like buildings, so that you will have a rich feeling. Some shops also you see, like clothes shop or even jewelry shops, they will put mirrors. So through the mirrors, when you are reflecting and seeing too many objects, then you will feel the shop is much bigger and much vast. So many a times this mimicking RNA also will do the same impact, the same effect to the system. Actually, it is a mimic. It can also sequester some of the proteins where the actual RNA is supposed to be pairing, but the levels of this mimic RNA make sure that the actual RNA level is also kept in check. So such kind of mechanism do exist. So the another non-coding RNA is Xist, which we have studied properly. That is helpful in the X chromosome inactivation, and it is initiated by X chromosome inactivation specific transcript, and it occurs only once during the embryonic development and at around 4.5 to 5.5 days of embryonic development. And this time the X chromosome is maintained as X inactive during the future cell divisions. Since X chromosome inactive maintenance does not depend on Xist, it was always assumed that Xist should serve no purpose subsequent to initiation of X chromosome inactivation. However, what we know that the Xist is continuously expressed for the entire life of the female. This suggests that Xist serves as additional functions apart from what we know. After the X chromosome inactivation is established, the Xist continue to be expressed with a purpose. So a recent study in mice has shown that the X reactivation resulting from the Xist deletion leads to development of a highly aggressive lethal blood cancer. So what we understand, the Xist non-coding RNA have other functions also. So this is called mixed MPN MDS. In this also creates a multi-drug resistant type of cancer in females with 100% penetrance. The penetrance and expressivity is a genetic term. Probably you have already heard. The penetrance basically sees whether or not you have the availability of a given gene. Expressivity means what is the level of expression of that gene based on what you have got. That also can vary. So if it is just like you have a pen and paper. If you have 10 people and if you give pen and paper to them, they may or may not utilize it. One thing, that is penetrance. And expressivity means some will write a letter and some will make a beautiful art form and some will just scratch on the paper and ruin it. So this is expressivity. So this what you say here is that Xist here continuously expressed for the rest of the life of the female, and we know from a study that the X reactivation can result from Xist deletion, because reactivation is happening in the absence of Xist indicates that Xist was needed throughout the life, and this leads to the development of, so X, if it is reactivated due to the absence of Xist, it can lead to a highly aggressive cancer, and which is 100% penetrance means no one is exception. Everyone has the same phenotype. So the upregulation of X-linked genes resulting from the deletion of Xist leads to changes, because if the X chromosome is activated, then gene expression also happens from this active X chromosome, and they are important. They have they cause this aberrant activation cause changes in the homeostatic pathways leading to cancer. Many a times, many homeostasis pathway does adequate cell proliferation, adequate cell metabolism, adequate free radical production, etc. But when you have more than adequate, more than required, then they will overdo it. It is just like you will chew your mouth if food is there. So you are biting your both the jaws with the food in between. But if you constantly bite without the food in between, what can result? Your enamel will erode. Eventually your teeth will erode. Your teeth is not supposed to grind against an equally strong material. You don't take stone in your mouth and grind it. If so happens, your teeth will be short-lived. So you cannot have a long-lived teeth. Same logic applies even if they are housekeeping pathways. If they are overly expressed, if it is overly doing, that can lead to situation like cancer. So this is consistent with the non-association of supernumerary X chromosome and several human cancers. So we should understand that the non-coding RNA, no matter whether it is important for maintaining the homeostasis or it is a essential molecule that allows the cell to survive from a juncture, or this non-coding RNA is a decoy for another non-coding RNA, irrespective of the actual role played by this non-coding RNA, what actually happens is that it can influence the normal functioning of a cell. Sometimes it can lead to cancer and sometimes it can lead to aberrant apoptosis. Either extremes are possible, and we have seen several examples of non-coding RNA that is governing the homeostasis and sometimes leading to formation of tumor. So we will move on to a new topic, that is maintaining of the telomere, and enzyme responsible for that is telomerase. So telomeres and telomerase are an enigmatic and yet well studied, yet too much important for it is for the normal survival of a cell. So we can say it is something like needed but not needed. So what we should understand, the telomere is an essential feature, but its shortening also is an essential feature. So although shortening is, it's just like, human, if someone says humans or any species death is essential, you may wonder, oh, what this person is saying. But actually you can imagine if each and every organism on this planet do not die, there is no scope for variety, no scope for new species, all the nutrients and minerals, everything will get trapped in some organism. So plants also has to die, animals also has to die, even bacteria also has to die. Every organism appear, reach its peak and disappear. So this is important. So technically death is a way of recycling the nutrients, minerals and various other materials you accumulated from this nature. So that has to be released back. So that is possible only through death. So telomere and telomerase also something like that. So telomere maintenance is done by telomerase, and telomerase is an important molecule, but if telomerase is present everywhere, it becomes unimportant molecule or useless molecule, or you can even say it is a harmful molecule, because cells also have to die at some time point, and that death is with a purpose for new cells to form. It's just like wildfire. If someone asks, is it wildfire important? And everybody will say, no, wildfire should not be there. But in nature point of view, wildfire, it may appear cruel, but once a wildfire is done, there is scope for lot of new plants to grow. It will make the ash is lot of rich in nutrition. Same with a volcano eruption. Volcano eruption kills lots of living organism, but the nutrients it brings in makes the volcanic soil is rich in nutrient and the plants can grow nicely. So telomere and telomerase has to be seen not just like a scientific way, it also has to be seen in a philosophical way. So as the background, we have to ask, what is telomere and why do we have them, and how do they get there, and what do they do, why do you care about them? And you can see a lot about this telomeres in this Nature Structural and Molecular Biology article by Gao et al. So it's a quite interesting molecule. What is telomeres? Telomeres first described in 1975 by Elizabeth Blackburn at Salk Institute for Biological Studies, who recently in around 2009 earned Nobel Prize for her discovery because it is so important. So they are repetitive DNA sequences that ends all chromosomes of every eukaryotes who have got a linear DNA structure. Our chromosomes, each chromosome is a single linear DNA structure. It has got two ends. So they contain thousands of repeats of 6 nucleotide sequence T2AG3, TTAGGG. It is 11 kb at birth to 4 kb in old age. 11,000 bases are there at the time of your birth, and as you grow old it becomes around 4 kb in length. In humans there are 46 chromosomes and 92 telomeres, 46 into 2, one at each end. So the telomeres protect the chromosome from the replication related loss of vital genetic information. We will learn more about telomeres in the next class. Thank you.